All right. Hi, everybody. We're going to continue where we left off on the macromolecules presentation. Tonight, we're going to talk about nucleic acids, and that'll take us to the end of macromolecules and the end of the unit. Woohoo! There's a few review slides at the end, and I'll maybe go over them if we have time. All right, so nucleic acids is what we're going to be talking about now. Nucleic acids, their function is to store information. I think that alone is freaky. That we have biological molecules in our bodies that store information in the same way that, like, a computer program stores information. I think that's crazy. So, nucleic acids are made of several different atoms. We've got elements. We've got carbon, hydrogen, oxygen, nitrogen, and phosphorus. We are not seeing phosphorus in very many others of the biological molecules. You see them in phospholipids, and you see them in this phosphate group right there. So just be aware of that if you see phosphate or phosphorus, that's an indicator that you might be dealing with a nucleic acid. So there are two types of nucleic acids. That's DNA and RNA. So DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. And you're probably familiar with DNA, but you might not be familiar with RNA, so we'll learn about that. The polymers are the nucleic acids themselves. If I get my picture out of the way, you can see that this is built of a whole bunch of nucleotides. The nucleotides are the monomers. Those are the building blocks that make up DNA or RNA. If you look at the nucleotide structure right here, it's got three main parts to it. And I would say that this is one of the easiest ones to really stump people when they're trying to identify their molecules by looking at them. They get frozen here when they're first learning them. So each nucleotide has three parts to it, and they're always the same. One part is the phosphate group. And when you see the phosphate group, it's often drawn as a circle. So if you look down here, you can see the P, and it kind of has a circle around it. That's the way they draw the phosphate. The second part is the pentose sugar. Pentose simply means it's a five-sided sugar. In DNA, the name of the sugar is deoxyribose. In RNA, the name of the sugar is ribose, and they're slightly different, and it's showing you the slight difference in those two. And then the nitrogenous base is up here. What the word nitrogenous simply means is it has nitrogen in it. Now this one, so let's be clear. This is a carbohydrate. It's a monosaccharide. So when you look at it and you're trying to identify what it is, you'll be like, oh, that's a monosaccharide. But then up here, here's two fused rings. And we know that those can be found in lipids in steroids. And so it's just a head scratcher sometimes. So the dead giveaway should be the phosphate group. And then you should go from there and try to just kind of memorize what the different parts of the nucleotide are. Okay, let's go ahead and move on. I'm trying to see if there's anything else in this that you would want to know. I think we're good. Moving on. All right, so DNA is named for the sugar in its nucleotides, which the sugar is called deoxyribose, and it stores information. We already learned this. You might not remember it. It stores information about the primary structure. That's a technical term, remember. The primary structure for proteins. So remember, there's primary, secondary, tertiary, and quaternary. DNA doesn't say anything about secondary, tertiary, or quaternary. It teaches you about it tells the cell how to build the primary structure of a protein, and that's simply the order of the amino acids. So the DNA says, put this amino acid, then this amino acid, then this amino acid. It also stores information about the sequences for RNA molecules. So we're going to learn what RNA is in a little bit. I will tell you that we're going to have an entire unit that's all about DNA. So right now we're just introducing you to the molecule itself. We're not teaching you really how that molecule works or how DNA and RNA work. The other thing that's super important about DNA, crazy important, and RNA is that it's heritable, so that you inherit it from previous generations, from your parents. All right, and then just a quick little touch on history, because it's interesting and it's a little bit controversial. These two fine fellows on the left received the Nobel Prize in Science for discovering the DNA double helix shape, which simply means a double helix is like a ladder that has steps going across, but then the helix indicates that it's rotating, so it's constantly turning. And they received the Nobel Prize for that. However, they were using Rosalind Franklin's X-ray crystallography technique that she had invented, so they kind of used her work in order to make this discovery, and she did not receive the Nobel Prize. So some people are concerned that maybe she didn't get the Nobel Prize because she was a woman in that era. However, she also happens to have died prior to them receiving the Nobel Prize. So there are some who say, well, she didn't receive it because she had passed away. Sadly, we don't know exactly why she died. We know that she had ovarian cancer and that she died from ovarian cancer. But if you think about, she was extremely young. I think she was 34, making stuff up. I don't know. I think she was in her 30s when she died. But she was working with x-rays, and so there's question, is it possible that her work with radiation is what led to her really early onset cancer, or would she have developed early cancer as a young person anyway? We don't know that. Okay, so moving on, DNA structure. Obviously, a DNA molecule makes that double helix, and that picture is right above my picture right there. There's a DNA double helix. All right, so there are 4 what we call nucleotides or nitrogen bases in DNA, and I'm going to interchange those 2 terms right now, but they are not technically interchangeable. So take a look at the diagram here. This is a DNA molecule, so that the double helix looks like this. If we untwist the double helix and then look at the steps on the ladder, that's what we're looking at right now. And if you look, the nucleotide, remember the phosphate is always shown as a circle, and then the 5-sided sugar is right here, and that sugar is deoxyribose. And then attached to it is the nitrogenous base, or the nitrogen-containing base. These are the bases that could be one of 4 choices: adenine, thymine, cytosine, or guanine. Sometimes we will refer to a nucleotide, which is the whole thing, as adenine. And that's because this part is always the same. Adenine is the only part that's changing, or thymine is the only part that's different between the different nucleotides. But technically, when you're talking about A, T, C, or G, you're just talking about this little part of each nucleotide. You're not talking about the sugar and the phosphate, but they're somewhat interchangeable. All right, so DNA has these 2 sides to it, and they are bonded in the middle by dotted lines. And hopefully you're starting to recognize that when you see dotted lines, those are hydrogen bonds, and they're dotted to indicate that they're weakly bonded. So what that means is the DNA double helix can untwist, and then when the DNA is getting ready to be copied so that your cells can divide, the 2 sides can actually peel apart from one another because those hydrogen bonds are really weak. And then we'll learn later about how they copy each side. Super important part of the DNA structure is that the certain nitrogen bases are always across from each other. And look at the diagram and note that as you're looking at it. So adenine and thymine are always across from each other, and cytosine and guanine are always across from each other. So find that on the diagram. So on this diagram, they're showing you the phosphate sugar, phosphate sugar, phosphate sugar backbone. On this diagram, it's the same thing, but they turned that phosphate sugar backbone into just a solid ribbon. This again is the same diagram, only now they're showing it twisting. So they're just showing the rungs on the ladder, the nitrogen bases, as different colors. All right, the backbone, which is made of sugar phosphate of each chain, consists of nucleotides that are covalently bonded, which are strongly bonded to one another. So while the 2 sides of a DNA molecule can be unzipped, they can be pulled apart because they're just weak hydrogen bonds holding the 2 sides together. The backbone itself, super strong and should never come apart. And we'll learn more about that in our DNA unit. Oops, I didn't click that right. Hold on just a second. There we go. All right, let's talk about what RNA is. RNA is related, but it is more disposable. It is less stable, and your body spends less energy working on it. So just really quickly, the way that I describe this, DNA is like the originals that an architect has made for a building. So, there's blueprints and that kind of thing. A blueprint is a copy of the originals. So DNA are the originals. That's like the computer in the architect's office, and it's super important that we have that information and never lose the actual design for a building. But the RNA, they're like the blueprints. So they're like the copies. The architects office. They print off the blueprints, they send it to the construction site, and the construction workers are out in all kinds of weather. And sometimes those blueprints get torn and rained on and crapped on and whatever. Does it matter? No, crapped on. Who knows? Does it matter if they get ruined? No, because the architect's office can always print another copy of them. So that's what RNA is like. We keep DNA tucked safely in a nucleus, and we try to protect it at all costs because we don't want anything to screw it up. And then it sends out messages that are called RNA. And if the RNA gets damaged or it falls apart in the process, the DNA in the nucleus can just send out another one. No big deal. All right, so RNA functions to carry messages from the DNA to the ribosome, we'll learn what that is, about how to build a protein. They have many, many other functions. We know of at least 15 different types of RNAs. You're going to need to know 3 of them, but not yet. So just if you think of them as messengers, we're good enough for right now. They are less stable than DNA. The cell expends lots of energy to make DNA as stable as possible. That's part of why it has 2 sides. And there's other things that I'll tell you about. That's why it has a special sugar, and it's why it has a special nitrogen base. But the cell's willing to conserve energy on RNA because it's not as important. So if the RNA gets damaged, we'll just make another copy of it. So it's less stable than DNA. So let's take a look at the DNA and RNA structure. So before I say anything, you should already know the difference between DNA and RNA. So if you look at the DNA, you see a double helix. It's got 2 sides that are twisting. The RNA is single-sided. It only has 1 side. The name of the sugar in RNA, we already said DNA's sugar is deoxyribose. So the sugar in RNA is ribose. How else are they different? RNA is single-stranded, and notice that it has a nitrogen base called uracil instead of thymine. So uracil, why does it have uracil instead of thymine? Thymine takes, we think this is why, thymine takes more energy and it's more stable. So it takes more energy for you to build DNA using thymine. But your body can conserve energy and use uracil, but uracil is a little less stable of a molecule, so it's possible that it would fall apart. We'll just build another one because we've been saving energy. All right, so this is called the central dogma of molecular biology. I hate that name, but it is what this is called. The central dogma of molecular biology is the idea that we start with DNA. All information is stored as DNA. It is transferred to RNA through a process that's called transcription. We're going to learn about transcription later, not yet. And then that information is used to build proteins. So the information stored in DNA is transferred to RNA before the protein is built. That's called the central dogma of molecular biology. Like, that's a phrase that I think, is so not interesting. That's what they call it. So there you go. All right, that's it. We're done talking about the nucleic acids because we're going to have a whole unit on DNA, so we're not going to spend a lot more time on it. These are some things just for reviewing for macromolecules in general. Please make sure you can identify the structures of the monomers and polymers of the 4 major classes of macromolecules. However, lipids don't really have monomers or polymers. Like, they're not building blocks, they just are. Whereas polysaccharides, their monomers are monosaccharides. You should be able to diagram the synthesis, so dehydration synthesis, and hydrolysis, that's the opposite, that's adding water to break molecules apart, of carbohydrates and polypeptides. You should be able to explain the functions of all of the molecules discussed in this presentation. What is the function of a carbohydrate? Well, they are quick energy or like short-term energy storage. Lipids are long-term energy storage, just as an example. Explain the emergence of all 4 levels of protein structure. The emergence is an intimidating phrase. Just understand what are all 4 levels of structure in proteins. If you can explain how they work together, you're good to go. And then it says, describe the general role of nucleic acids in living systems. Their function is information storage, and you're good enough on that one. Identifying macromolecules, these are things that you should look for when you're trying to recognize pictures of them. The carbohydrates are often cyclic, so they look like hexagons or pentagons. I call them rings, but they can be linear. Remember, a ring can be pulled apart into a straight line. They have carbon, hydrogen, and oxygen in roughly a 1 to 2 to 1 ratio. That's really important because the lipids are really, really, really long chains of carbons and hydrogens, and so you can easily think that they are a linear carbohydrate. But a carbohydrate has way more oxygen. So in glucose, for example, it's C6H12O6. So 6 O's. Whereas in a lipid, you only ever have 2 oxygens. You would never have, or in the I shouldn't say in all lipids. In the fatty acids, you would only have 2 oxygens, and they're both found in the carboxyl acid functional group that's at the one end that determines, that gives the fatty acid its name acid. All right. Sometimes P if the lipid is a phospholipid. So sometimes you'll see phosphorus. And remember that a phospholipid, we draw it like a circle with 2 tails coming off of it. The circle is the phosphate head that is hydrophilic, and then it has the 2 tails that are hydrophobic, and that's found in cell membranes. Proteins, I should have said something more with the lipids. Don't forget that the steroids are fused rings. I should go back and change that. Proteins are long chains of amino acids. Amino acids have an amine group on one end and a carboxyl acid group on the other end. They include carbon, hydrogen, oxygen, and nitrogen, but sometimes we will see sulfur and selenium, but never do we see phosphorus in a protein. So that's a way, if you see phosphorus, you should be like, that is not a protein. So the nucleic acids, that's what we just covered. DNA is a double helix, RNA is single-stranded. The nucleotides are the monomers themselves, and they include rings, which can be intimidating. So the elements that they include are C, H, O, N, and P, but never sulfur. All right, and then I just have this little thing at the end, the functions and identifying. Can you identify these things, and what are they, and then what's their function? And then I will mention at the very end, I have this link to something I made ages ago. It's a little purpose game, and I made it actually for honors biology, so it's not perfect, but it's pretty good. And I can't remember how many people have played it. 13,000. It's been played 13,000 times. Just saying. So I can make a better one. I just haven't had time to make a better one. But you might want to try this game. You press play, and then it tells you, so identify the polypeptide. And so you look through here and find the polypeptide. Blah, blah, blah. Have fun. That's it, you guys. Have a good night. Bye. Oops. I don't know where the link is to shut this down.